This is the Introduction to Nutrient and Gene Interactions lecture from earlier in the year, and the topics covered here are meant for you to apply to some of the other topics you're going to get now as we move into the module a little bit deeper and cover some of the additional topics in nutrition and health. So the learning outcomes are for you to review the processes involved in gene expression, understand and be able to apply the key terms to other topics covered in this module and in other modules that you're taking in your program. Understand working examples of nutrient-gene interactions that have implications on health, and develop an ability to recognize when various topics in human nutrition and metabolism have a component which might be actually driven by a nutrient-gene interaction. So this is a very simple schematic outlining the processes that are involved in gene expression. So proceeding from the DNA through to RNA into a translated protein. Now, I'm not going to cover this information actually in this lecture, but you can click this link here towards the bottom of the slide, and this will take you to a nature.com website, which has a lot of information at the appropriate level for you to review and understand the process of gene expression. There are also additional terms in at that same website, which will take you through very nice scientific definitions and working definitions of the following common terms that you're going to hear when you look at this topic, either in textbooks or when you're reading papers, and understanding this glossary or these terms is very important for your ability to then actually reproduce the information that you've learned in exam question, answer situations, or in essays. I am going to draw your attention, though, to one thing relating to some of those definitions, and that's the genetic code. And this little diagram outlines how each amino acid used in protein translation has a distinct codon that relates to the messenger RNA and is essential for the process of gene expression and protein translation. What I want to draw your attention to is the fact that when we get into some of the issues that surround nutrition-related diseases that have or nutrition conditions which have a genetic component, that when we start speaking about polymorphisms or genetic mutations, it's generally referring to changes that occur in these codons. So it's these individual bases that are translated from the DNA to RNA, and errors which occur then produce mutations, which can then be result in the translation or the not translating a particular protein or having a protein with altered function, whatnot. So just so you're aware that this is what we'll be referring to, and it really comes down to functionality of the protein in most cases. At least the examples we're going to use in this lecture, and here is the examples, the list of the amino acids which are found in proteins that we synthesize. So just finishing up on that topic, genetic coding. There are 64 triplet nucleotides, and they're all called codons, and each codon encodes for one or of the 20 amino acids used to synthesize proteins. And there is some redundancy in the code in that there is more than one codon for a particular amino acid in some cases. Some other key terms that you need to understand and understand that there's a distinct difference in them is the term allele versus polymorphism. So an allele, a longer definition can be found here, this link, but is basically a gene variant that arises through gene mutation, DNA change, and it's preserved and conserved in the population. So you may have an allele that's a minor allele, or you may have an allele that's a dominant allele or a major allele for a particular trait that you express, and this is normal, and these are retained in the population through the production of offspring. Then there's polymorphisms, which are individual changes to DNA which produce, or which can produce, in some cases, changes to the alleles. However, they may produce no change, no visible change, or they may produce something that's not usually observed in the allele combinations that we have for some of the gene variants in our population. And then a better definition of polymorphisms can be found right here. So the term most commonly used, or you'll most likely come across in papers or book chapters, is going to be single nucleotide polymorphism. And this is when there's been a single base substitution. So at the genomic level, or at the level of the DNA, it will be described as 76, A for C, which denotes that the base adenine at nucleotide 76 was changed to a cytosine. This can also be expressed at the protein level if this polymorphism happens to be in the coding region, coding for a particular codon and an amino acid. Then you get at the protein level, you could have W26C, which denotes that the tryptophan, that amino acid 26, which is a tryptophan, is changed to a cysteine. Gene mutations are alterations in the DNA sequence. So natural, spontaneous mutations are changed by cellular processes, which end up being a miscopy of DNA. This can also occur by oxidative damage to the DNA. So to induce a mutation, something needs to occur in the cell's environment. And the cell's environment can be affected by things such as diet, chemicals, radiation, viruses, lifestyle. And most mutations, though, are actually repaired, and there are very few errors that escape the DNA repair system. But there is a percentage, because we replicate, our cells replicate at such a high rate, and turnover is such a continuous process throughout life, that errors do occur and sometimes can be retained in the organism. However, these mutations are not necessarily always harmful. They can also be helpful, and this is the process of natural selection that we associate with scientists such as Darwin. So this just gives you a schematic overview of the different combinations of alleles that can occur. So you're homozygous for a particular allele. That means you have, for both copies of the gene in your DNA, you have identical genetic copies. However, if you have a substitution of a particular base, you may have it in one versus the other of the two genes, and then you'd be referred to as heterozygous. So you'd end up having two different alleles. Or you could be homozygous for a minor allele, or a variant, or a mutant, which is the least common in the population generally. So, and then this would be defined as having two copies of the gene in question having different bases compared to the majority of the population. So in summary, some genetic terms that you should have a good understanding of would be gene, being the length of DNA that encodes for a particular polypeptide. A genotype, the combination of the two alleles that appear in a pair of chromosomes. And the phenotype, which is actually the observable physical or biochemical trait that an individual expresses as a result of the particular alleles that they have. In terms of diseases, these are examples of single gene diseases where we know there's a single gene affected, either by mutation or by several mutations, that produces a disease that has a known pathology. So these are diseases such as cystic fibrosis, muscular dystrophy, familial hypercholesterolemia, or phenylketonuria. But most diseases, when we talk about nutrient-gene interactions, or the diseases that arise from a nutrient-gene interaction, tend to be complex diseases. And so these generally are influenced by two or more genes, and with possibly or most likely environmental influence as well. So that's what we would describe then as complex inheritance. So that's when you inherit a particular gene or several genes, and your disease risk or your disease outcomes are really dependent on the particular genes that you have, but as well as the environment you live in. Polygenic diseases, this is when you have genetic changes to a large number or a group of genes, but these changes are small and minute, but collectively, if you have the right combination or collection of these particular genetic changes, they could have a significant impact on the final outcome or condition of a particular disease. Then there's multifactorial disease, and this is where environment plays a larger role, where you can have an interaction between multiple genes and multiple environmental conditions. And so these can be pollution, so air pollution that we experience even in the cities that we live in may interact differently with some certain individuals' genetic predispositions. It could be related to cold environments, it could be related to hot environments, it could be related to other diseases or disease vectors such as things like malaria. And then there's continuous or quantitative traits, and these are things that are measured in the population and occur across a spectrum or a normal distribution. And people in a particular area of that normal distribution, so whether that's BMI or plasma cholesterol levels, tend to be at higher risk of a particular disease or at lower risk of a particular disease because of a combination of some of their diseases. And continuous variation, so height would be an example of this. So this would be basically, this is an estimate of the average UK height of an average UK male. And so you can see there are very few people under 160 centimeters and very few people over 188, and most people fall between that bracket. Well, for polygenic diseases, it's the same thing. So if you look at several possible alleles in the population and having a particular combination of those alleles, you can see that there's very few individuals that would have the recessive versions of the alleles versus, and have them in a combination where that they have complete recessive expression across the board, as opposed to, on the other end side of the spectrum, people who have only dominant expression, and generally the rest of the population falls somewhere in the middle. And so this is basically what we look at as continuous variation when we look at it from a polygenic disease perspective. So there's also common diseases that cluster in families, which again gives us a good idea, or it's a good indication of whether or not it's a genetic disorder, as well as whether or not it's polygenetic and what influence environment has. Because, so diseases such as heart attack and stroke or breast cancer, these can cluster in families, but it's not necessarily a given that you will develop one of these diseases just because somebody in your family has. There's an increased likelihood, so your risk tends to be higher. But remember, families not only share their genes, but they also share common environment, culture, behavior, diet, all these sorts of things. And these all have impact as well. So genes in the environment and how this interaction occurs and what this interaction actually means. Well, there's constantly interaction taking place between genes and environment, and this will invoke a particular cellular response. And so you got to remember too, for environment, there's the environment of the organism, so where you live, the food you eat, the atmosphere you breathe. This is for the organism as a whole, but then there's the environment of the cell, and the cell's environment is much different, and it's basically what's in the cytosol, what's in the extracellular fluid, and the factors that affect this particular cell in terms of cell signaling and other nutrients or chemicals that reach the particular cell. So environment is a generic term that refers to everything from the environment of the organism all the way down to the environment that the cell finds itself in. And when this particular environment, whether it's at the organism level or at the cellular level, affects gene expression, well, then this is what we can say is an environmental interaction with the genotype. So, and then this can have an effect on phenotypes and not necessarily in an understandable or predictable manner. So things like obesity, type 2 diabetes, cardiovascular disease, these are all under control, or sorry, hypertension. These are all controlled by different genes, and how these genes react to a particular environment will dictate how the phenotype is expressed. So this is looking at it on a spectrum. So if you wanted to put disease or causes of death in humans across a spectrum, you would have those on one end, which are totally genetic, and these are things like cystic fibrosis or muscular dystrophy. But then you have the spectrum that occurs where you have a lot of other diseases which have a gene interaction related to them, but are also directly affected by their environment. And this is where a lot of these diseases fall. And then you have things that are totally environmental. So that would be like a motorcycle accident or a motor vehicle accident, being struck by lightning, being poisoned by a chemical spill, something along these lines. These are totally environmental and not related to your genetics. There's also the concept of penetrance. So a highly penetrant allele variant directly causes a disease. So this is something like Huntington's disease or cystic fibrosis, muscular dystrophy. If you have the particular allele variants for those diseases, you will develop those diseases, and that's what it basically means. The equation is a very low chance, low risk of you developing one of these diseases if you don't have the particular variant that causes the disease. But most common diseases are polygenetic, and a given gene will have limited penetrance for a particular disease trait, and therefore the environment is very important. And one of the things that influences a cellular environment is diet, physical activity, and also physical states, so things like diabetes, things like obesity. And so when you take these into account, they will have major influence on the phenotype that we observe. And so this is just a schematic explaining that. So you have the gene-gene interactions and the gene-environment interactions, and these all contribute differently towards what actual disease phenotype looks like. So looking now then at nutrition and the human genome, there are two ways that nutrition really relates to the human genome. The two terms you may have heard, which are not interchangeable, are nutrigenomics and nutrigenetics. And the effects of a bioactive compound in nutrition on gene expression and the resulting changes in physiology is basically the study of nutrigenomics. So this is how the bioactive compounds actually interact with the genes to change the phenotype. But then there's the other side of this study, and it's where you look at genetic variation and how inter-individual differences in responses and the implications for susceptible subgroups can occur in the population. And this is where you're studying nutrigenetics. So this is when you're looking at the genetic variation, knowing that this genetic variation will have an impact, will be impacted by nutrition in some way. So to look at it from a study perspective, this is how it looks. Nutrigenomics takes into account all the other omic technologies that we have, and then we can measure if you feed somebody a particular food or remove a particular food from an individual. You can measure things like the transcriptomics changes, the proteomic changes, the metabolomic changes, and these are changes in things like the expression of our specific RNAs, expression of proteins, the production of metabolites, and these all affect the phenotype. And so this will be the nutrigenomic approach to studying nutrient-gene interactions. But then there's the nutrigenetic approach, which is where you look at a particular food or bioactive and how it interacts directly with the genotype of the individual, and then how that subsequently affects the phenotype. So for diet-gene interactions, there's also some distinctions that should be made, because you can have a particular genetic polymorphism that can influence your risk factors for a particular disease and the expression of that particular disease, but that it can be completely independent of the diet, even if that particular disease does have a dietary interaction or link. But polymorphisms can also interact with the diet itself and modulate risk factors for disease. So some examples of that are following in the next couple of slides. So an example of that could be body fat and the distribution in the population, where body fat is equal to your genotype as well as your environment. And so this graph just gives you a cartoonish, but illustrates that if you have a particular genotype, say genotype A, you are automatically going to, because of this genotype, independent of what food you eat, have a higher body fat content. However, your starting point, or not your starting point, but the lowest point at which you can reach in terms of your body fat percentage is probably capped for this particular genotype. But you can influence weight gain or more fat increases based on the amount of energy or fat that you consume in your diet. And subsequently, there are other people in the population who tend to be at the other end of the spectrum. Their genotype protects them in some way from excessive body fat accumulation, but they too are susceptible to the nutritional drivers, which are energy and or fat intake. So a working example of this is polymorphisms that occur in the ApoA5 gene. So ApoA5 mediates the hydrolysis of triacylglycerides and VLDL particles by lipoprotein lipase. And there are variants associated with this particular protein, and they relate to tag levels. And the paper cited here, Cordelia et al., 2007, investigated this interaction. And so specifically looking at this particular polymorphism and linking it to fat intake in relation to BMI and obesity risk. This was in quite a large group of people, so this is about 2,200 people, both men and women. And so what they showed was, in this particular study, that when monounsaturated fatty acid intakes were less than 11% by total energy, BMIs for people who had the TC and the CC, or the non-dominant, actually I'm not sure if this was dominant or not, but if you had the C allele, your BMI was increased more than that of individuals who did not have the C allele. And vice versa, when monounsaturated fatty acid intakes were greater than 11%, the BMI of the individuals who had the C allele was actually less than that of those who had the T allele. So this is the figure from the paper which demonstrates this. So low MUFA intake, BMI of the C carriers had a higher BMI than those with the T allele only, and vice versa. At high MUFA intakes, the BMI of C carriers was lower than that. So this is demonstrating an interaction, a genetic interaction with this particular genotype that has a direct effect on BMI and likely on body weight. So some of the rationale behind it is that maybe this is linked to how these individuals metabolize certain fats, possibly in the postprandial period, and what happens to the fats if they're not lowered from the plasma at a fast rate. Or these may be also related directly to the protein that ApoA5 interacts with, or the enzyme, which is lipoprotein lipase. Another good example of a nutrient-gene interaction that has an effect on a phenotype would be methylene tetrahydrofolate reductase. So this is the enzyme involved in homocysteine metabolism. And it catalyzes, this enzyme catalyzes the reaction of 5,10-methylene tetrahydrofolate to 5-methyl tetrahydrofolate. And 5-methyl tetrahydrofolate methylates homocysteine to methionine. And that reaction is catalyzed by methionine synthase and involves the cofactor B12. And this is very important to one-carbon metabolism in our bodies, and this is a key regulatory step in some processes related to cell division, as well as I said, producing those one-carbon intermediates that are required for various other metabolic reactions. There's a relatively well understood now polymorphism which occurs in methylene tetrahydrofolate reductase, where you have a C for T inversion or switch, and the T allele actually causes reduced activity of that particular enzyme. And that's associated with a fall in the plasma concentrations of 5-methyl tetrahydrofolate, which is basically the active form of the vitamin folate. And this slows the conversion of homocysteine to methionine. And it has another knock-on effect where that you increase the plasma concentrations of homocysteine by about 20%, and you eventually start to see homocysteine appearing in the urine. Now, there have been a number of meta-analyses which have linked these higher levels of homocysteine to heart disease, but this link is not a causal link, because those studies have been done as well to show that there's no direct link here, but in terms of causation. But there is something occurring in this particular metabolism and disruption of this particular metabolism, and namely the production of methionine from homocysteine, that has that in some way, shape, or form affects coronary heart disease. So the TT genotype is associated with increased plasma homocysteine, and only in the individuals who have low folate status, because you can get around this particular polymorphism by consuming higher levels of folate, obviously, because you're still supplying the cofactor that's required for the reaction to convert homocysteine to methionine. There's no direct association between the TT genotype and CVD in terms of a causal link, but there is a link there, which we don't fully understand at this stage. So dietary interventions. Should individuals who are homozygous for the 167T allele be identified and measures taken so that they consume adequate folate? Or do you just target the entire population to make sure they're covered, make sure everybody has adequate folate intake? And these are some of the strategies that people look at when they're trying to, as these diseases are being understood or being discovered, and we're trying to understand what their implications are on health. Because there are negative sides to this coin as well. Not everybody needs high dose folate, and it can have other issues in the population, like masking pernicious anemia and such things like that. So this is always a question that is very difficult to address from a public health perspective, but nevertheless very interesting from a scientific perspective. So the next disease I want to talk about is another very rare disease in the population, but one that does have a very distinct genetic component to it, and that's cytosterolemia. So if you have a mutation in one of the two proteins referred to here, A, B, C, G5, or G8, these are ATP binding cassettes, G5 and G8. These are linked to sterol regulation in the body, primarily expressed in the gut at high levels to help prevent absorption of plant sterols in the diet. And these occur, their function seems to work alongside that of MPC1L1, which is the sterol absorption protein found in the gut. So this keeps levels of plant sterols low and cholesterol high. This is the scenario that we want, in the sense that we do not want too much phytosterol being absorbed and able to interact with our sterol metabolism. So cytosterolemia, as it says here, is an autosomal recessive disorder. It's directly linked to an unrestricted absorption of both cholesterol and plant sterol from the diet. Clinically, it presents as high plant sterol concentrations in the plasma. Individuals will develop tendon and tuberous xanthomas, so these will be on the knuckles in the back of the leg, say in the Achilles tendon. There is also accelerated atherosclerosis and premature coronary artery disease, usually resulting in death at a young age. So maintaining hepatic sterol levels is very important, and you can see here from an animal study which looked at knocking out this enzyme, so artificially disabling this enzyme, that these particular mice, they had excessive amounts of plant sterol present in both their liver and their plasma, and the knock-on effect that this had was to result in a very decreased amount of bile being produced. But you can see that overall the total sterol concentration in these livers didn't change. So the excessive amount of plant sterol coming in from the diet had several effects. One, lowering actual cholesterol levels. Two, severely affecting bile acid. And three, increasing plasma concentrations of plant sterols. So as a result, the liver tends to have a compensatory response by effluxing cholesterol out of the liver to maintain a constant sterol level in there. However, as I said, plant sterols are not something that we want interacting in our sterol metabolism pathways, because they offer no benefit and they can't be converted into the appropriate hormones or steroids that we require. Then from a different perspective, let's look more globally at particular genotypes that have developed as a result of environment individuals live in. And you can see here across the top of this graph, so this is from a paper published in Science a few months ago, that if you look at the global distribution of locally adapted traits, you can see individuals have been conserving some useful traits which likely arised either in the population by chance or as recessive or lowly expressed alleles. And these can be things like, as you see up here on the top, adaptation to marine diet, lactose intolerance, malaria resistance, etc. And you can see that these developed most likely independently and affects, while some cases affecting the same genes, in other cases affecting very different genes. So let's look at, for example, malaria resistance. If you look at it, how it's developed in South America, it's linked to a different gene than that of Africa. And then also if you look at cold climate adaptations, you can see here in Russia, northern China, Mongolia area, you have a very different set of genes that are affected compared to that of the Greenland Inuit. Adaptation to other things, let's see, such as arsenic, high arsenic levels in the environment, again, different genes being affected. So you can see that we can get to the same point or the same phenotype, but by having very different environment/gene interactions to drive us to that point. And just focusing a little bit on the Greenland Inuit and Danes, because this links to some of the lipid lectures that you've probably had from me so far. If you look at this historical data from the Greenland Inuit and the Danes, where we compare the high fish diet of the Inuit compared to the Western diet of the Danes, and you can see distinctly different patterns in the phospholipids of the platelets, where you have higher levels of omega-6 fatty acids versus omega-3s in the Danes, but the Inuit tended to have very close to a 1-to-1 ratio, across these particular fatty acids. And they have lower incidence of heart attack and strokes, lower incidence of high cholesterol, lower TGs, and they tend to have higher LDL levels. However, this study that I showed you the last infographic from actually was interested in these particular changes and adaptations to cold weather. And what they identified was that some of these changes actually affected the fatty acid pathways. And so this has kind of turned some of the data related to the health effects of omega-3s on its side, because if you look at these, in particular these Greenland Inuit, who a lot of these omega-3 supplementation studies were based on, it appears that they actually have changes to their genome which result in a decreased synthesis of some of these fatty acids that are lacking in the Danish diet or generally in the Western diet. So what this tells us is that the cardioprotective effects that we've been associating with high marine oil intake may in fact be more so related to the fact that these individuals produce less of these fatty acids endogenously. And so that's maybe why we aren't able to replicate the low cholesterol and low total cholesterol levels and the reduction in heart disease and other atherosclerotic diseases that we see in these populations when we do high dose supplementation of marine oil in populations that don't have this particular genetic makeup. So just as one final point, I would probably assign you a learning task at this stage, and that would be to identify through your own research another common gene-nutrient interaction, outline how this disease or condition presents clinically, identify the underlying cause. So is it an SNP? Is it an autosomal recessive condition? Outline what, if any, dietary interactions are occurring, and then describe the overall impact of the condition in terms of what it would have on the population, healthcare management. So whether or not this is more something that you deal with individually because it's such a rare prevalence in the population, or is this something that is at a high enough prevalence in the population that it actually has impact on healthcare systems. Okay, so that will be a good tool to use in terms of a study technique and to arm yourself with some with a good working example that you've worked yourself through instead of just getting from the lecture notes. Okay, that's all I had on nutrient-gene interactions. Remember, the concepts here are meant to be applied to the other topics that you're doing in this particular module.